Another promise of PFA was tissue specificity. Is it really tissue specific or cell specific? First, myocardial cells are quite susceptible to PFA, but this might not be the only explanation for preferential ablation of myocardial cells. Electroporation kills cells of many types, but maintains the extracellular matrix and preserves blood flow into the region. And this therefore opens a window for regeneration of tissue over 1 to 2 weeks. For example, nerves may also initially be ablated, but then the myelin regenerates after a couple of weeks. Similarly, esophageal tissue may acutely be damaged, but can regenerate as well. Cardiac cells, on the other hand, are incapable of regenerating. Increasing doses of PFA around or in close proximity to the phrenic nerve caused diaphragmatic movement that eventually modulated and stopped, suggesting temporary dose-dependent stunning of the phrenic nerve, but not permanent damage, since this phrenic nerve always recovered. We have seen PFA as tissue preferential, and the data from clinical application shows the peripheral safety of the esophagus, phrenic nerve, and no instances of PV stenosis. Furthermore, esophageal and other tissues may be protected by layers of fat. Fat is a very effective insulator against PFA. The small fat pad between the esophagus and the left atrium, for example, can decrease the field strength reaching the esophagus and therefore minimize the risk of damage. Fat insulation may also explain why autonomic ganglia are not effectively ablated by endocardial PFA delivery. Ganglionated cell bodies are actually very susceptible to PFA ablation, equal to myocardial cells, but when these ganglia are surrounded by fat, they are very difficult to penetrate from the endocardial surface.